Good day, and welcome to the SNN.Network Summer Virtual Event. Our next presenting company is Brooklyn ImmunoTherapeutics. It is now my pleasure to turn the floor over to your host, Howard Federoff, CEO of Brooklyn ImmunoTherapeutics. Sir, the floor is yours.
Thank you. Hello, everyone. It's a pleasure to be with you. Today, I'm going to be talking about the evolution and the future of Brooklyn ImmunoTherapeutics, a brief snapshot of where we've been and where we're going. The disclaimer is noted here for your reference, and let me just briefly talk about the company and its current status. We are now a clinical stage company, have been for a while, with a cytokine therapy in phase 2B, as well as a in-licensed, highly efficient nucleic acid delivery system, and as well a very efficient and effective cell reprogramming set of technologies, and in addition, then also in-licensed gene editing technologies, and I'll be discussing those in the moments to come. So Brooklyn ImmunoTherapeutics has a broad portfolio. I mentioned the phase 2B cytokine asset, which is currently being prosecuted in the neoadjuvant protocol for head and neck cancer. We have a team that has substantial experience and effectiveness in drug development, R&D, and healthcare. And we have a GMP manufacturing facilities and the capabilities on both sides of the company as we elaborate our future products. IRX2 is a cytokine therapy. It is ongoing, as mentioned, in a phase 2B setting, an evaluation of its potential effectiveness in patients receiving IRX2 compared to a second regimen, which I'll go through in a subsequent slide. There are many investigator-sponsored trials, which I'll also briefly enumerate in a variety of tumor types. And then there are 3 independent human studies, which in the past have demonstrated that IRX2 is able to activate, recruit immune cells following its subcutaneous injection. The new technologies are based on mRNA, and they enable gene editing as well as cellular reprogramming. So we have an exclusive license for a broad mRNA technology platform developed by Factor Bioscience. As I said, this is a mRNA cell reprogramming application, as well as gene editing, which can include gene editing not just in a cell and culture, but also, as we anticipate, in vivo, and a proprietary nanolipid particle delivery system. Our team is growing. We have recently recruited Kevin Demore, Dr. Demore, as our CSO, Ron Guido, our Chief Development Officer, Lynn Sadowski-Nation, our EVP for Clinical Ops, and Jay Seow, our Chief Administrative Officer. We will be announcing the recruitments of other stellar individuals to join our management team, but this is a very experienced and effective team. Our board is extremely seasoned and quite engaged. Charles Charrington chairs our board, Luba Greenwood, Dennis Langer, Eric Moore, and myself. This is a very experienced board with regard to biotech, as well as companies inclusive of those like ourselves that are publicly traded. Bear with me as the slide is just coming up here. We all love technology. Let me just go to the previous slide, which was skipped over. So we have, in addition to our operating team, the management team, and a great board, we also have constituted and continue to add members to our SAB, one for IRX2 is shown, Dr. Schnittman, O'Neill, and Wolf, as well as the newer additions, Dr. Andreeff, Dr. During, and Dr. Chris Rode of Factor Bioscience and formerly of Novellus Therapeutics. Our most advanced asset is the IRX2. This is a human-derived cytokine. As mentioned earlier, it is being pursued in a handful of ISTs in renal carcinoma, liver cancer, head and neck cancer, GI cancer, cervical and vulvar interstitial neoplasia, triple-negative breast cancer, and early-stage breast cancer. We anticipate that in the future there will be an additional phase 2 stood up as some of the phase 1 ISTs reveal the most promising clinical indications to pursue. We also anticipate that there will be a phase 3 study in the neoadjuvant head and neck cancer setting, which we expect to begin in 2023. We have a strong IP portfolio and patent position, and we have a cash runway that will take us through the end of 23. The human-derived IL-2, being one of the prominent cytokines within the IRX2 product, is able to have efficient interactions and activation of dendritic cells, T cells, and NK cells. With respect to dendritic cells, it improves their ability to be able to stimulate and ultimately present antigens, neoantigens, to T cells. It is able to promote in a neoantigen-dependent way T cell expansion and tumoral recognition and killing, as well as augmenting in the NK cell, NK context, the ability of NKs to recognize and kill tumor cells. IRX2, with respect to IL-2, when compared side by side, because this is a human-derived protein, it is normally translated, post-translationally modified, folded, and released, unlike some of the other products, recombinant IL-2, pegylated IL-2, and engineered IL-2. So it has few features that have been demonstrated both in preclinical, non-clinical, as well as clinical studies. The IRX2 is well tolerated. As I indicated earlier, in addition to IL-2, there are multiple other cytokines which we believe are activating networks that are important for immune recognition and ultimately margination and killing of cancer. Because of its normal conformation, it has greater functionality, as I'll demonstrate in the next slide. And as well, its dosing is more physiologically relevant than some of the pharmacological dosing that is required to see biological effects with some of the other forms of IL-2. One way to measure the activity of IL-2 would be to look at a displacement assay or measure it and sometimes report it out, as will be shown in the slide when it materializes, which is to show that IL-2 that is of a human nature compared to a recombinant nature is distinctly different. It is many-fold, if not a log order, more effective in this displacement assay, whether it's in YT1 cells or KIT225 cells. When one compares, as shown in blue, the human IL-2 compared to recombinant IL-2. And so one inference that we draw, given the safety and tolerability of IRX2 with its IL-2 contained therein, is that this may potentially mitigate some of the known off-target effects and toxicity that may be associated with higher doses of some of the recombinant forms of IL-2, as has been previously described in the literature. In the setting of its subcutaneous administration, as you'll see in a moment in the next slide, in the left panel, you'll note that the administration of IRX2 is able to potently convert a cold stromal bed with tumor into one that's quite hot, reflecting the margination of a lot of different cells into the environment that is then associated with then the anticipated downstream clinical benefits. You can see in the lower right-hand corner, just from our head and neck cancer clinical trial, just contrasting what is seen with the IRX2 group on the left, where one sees substantial amounts of infiltrating CD8 positive tumor infiltrating lymphocytes compared to the regimen that lacked IRX2. This substantial difference between the two regimens would suggest that the immune activation that is attending the IRX2 subcutaneous local regional administration will turn out to be important with respect to its correlation to clinical benefit. One other observation that's been made is that IRX2 has a substantial additivity with respect to tumor reduction size in a preclinical animal model, where one can look at the impact of IRX2 alone, a checkpoint inhibitor alone, or as shown at the bottom of the slide, their combination, which is highly effective, suggesting, as would have been anticipated, that there might be a means to evaluate clinically the combination of IRX2 with a checkpoint inhibitor. And as I covered in an early slide, that's exactly what is being done in some of the combination studies that I referred to earlier. Another bit of data as it relates to the design of the INSPIRE study. This is the neoadjuvant phase 2b study that we've been talking about, where we have a study fully enrolled and anticipated top-line readout in the first half of 2022. The regimen randomizes patients to receive serial IRX2 or not, and then ultimately patients will receive the standard of care and longer-term follow-up, and that standard of care can include chemoradiation or radiation therapy. The IRX2 has a very modest adverse event profile, given that some of these are often seen associated with other concomitants of having a cancer diagnosis and or receiving other therapies. So its safety and tolerability is something that I think speaks well of its use, certainly in this setting, but perhaps, as we discussed earlier, in combination. This is a slide denoting all of the studies that IRX2 is currently being prosecuted, the monotherapy studies shown on the top and the combination studies with a checkpoint inhibitor shown in the bottom. These data will be serially read out, and we anticipate that the biology in combination, as would have been expected given the nature of the additivity between a checkpoint inhibitor of IRX2, portends a greater benefit than would be the case with either therapy alone. Brooklyn ImmunoTherapeutics has established manufacturing capability. Many runs of IRX2 have been produced good clinical product. The in-house manufacturing and process development capabilities allow for us to develop, as would be needed, additional drugs that could reflect alterations within the cytokine content of the IRX2 product. We will leverage existing Brooklyn staff as well as new hires to establish new gene editing, cellular reprogramming modalities, and doing those under GMP manufacturing. Little or no outsourcing of our manufacturing increases the speed and reduces the cost of producing our product. So part of the history that I referred to earlier about Brooklyn's evolution is really shown on this slide, where one can see, as it will come up in a moment, then scanning from left to right, we made the decision and we in-licensed a substantial amount of intellectual property from Factor Bioscience and Novellus Therapeutics for novel mRNA-based gene editing and cellular therapy. The patents are well in excess of 100, and many more are percolating through the different jurisdictions with respect to their office actions and the like. The license is for the exclusive use of gene therapy and cellular therapies for liquid tumor types, solid tumors, sickle cell anemia, and other indications, including those of the monogenic type. Brooklyn made the decision and acquired Novellus Therapeutics a number of weeks ago, securing technology and the know-how to iPSC-derived mesenchymal stem cells in an unrestricted field of use fashion. That acquisition eliminates any future license payments to Novellus. It also transfers 25% ownership of Novocyte, which is developing an induced pluripotent stem cell-derived MSC, or an IMSC, for acute respiratory distress syndrome. This serial evolution, which has occurred just in a relatively short period of time since the company came public, will transform and is transforming Brooklyn to a platform company with numerous products in its pipeline of next-generation engineered medicines. Some of the core technology that attracted us, it ultimately was the basis upon our desire and successful execution of the licensing of these technologies, start with the upper left. It's the foundational mRNA cell reprogramming technology, where synthetic RNA directs the expression of a group of proteins constituting the Yamanaka factors to take a somatic cell to become a iPSC. Secondly, moving to the right, is an efficient mRNA editing capability where there is the ability to both delete, repair, and insert nucleic acid sequences. The modularity lends itself to the design and ultimately the execution of different types of proteins that have intrinsically different properties with respect to gene editing. Coming down to the right on the bottom, one such example is the context-specific gene editing protein with great nucleic acid sequence specificity, which is also one that is sensitive to the chromatin environment. So it's blocked by histone modification, which is probably accounting for why, when this approach is used for doing gene editing, there are far fewer off-target effects associated with its use. And then the bottom left is the lipid nanoparticle delivery system, Tornado. It can protect the RNAs from RNase. It is not inhibited by serum. It is able to effectively transfect both primary and induced pluripotent stem cells. And it can deliver mRNA cargo, both ex vivo in cells in culture as well as in vivo. And this, when it comes together at the center, really represents the basis of the construct of the license, which is our ability to develop a range of engineered clinical medicines, allogeneic, clonal, as well as other approaches such as that which I'll cover in a moment regarding gene editing, including in vivo gene editing. The value of starting with an iPSC to derive an MSC is one that is probably worthwhile spending a moment on. The fact that there is the clonal origin of the iPSC, which derives from a reprogramming, as shown here, of a somatic cell, such as a dermal fibroblast cell, can be done efficiently, and it even can be done concurrently if needed with gene editing to correct the mutation. This leads to a very well characterized mesenchymal stem cell at every level of characterization, which will address substantially the issue of identity, purity, and potency. If one starts thinking about the uses of these technologies, just a brief glimpse of what is in process for us, the non-gene modified iMSCs are going to be taken forward for bone marrow stromal rejuvenation in the setting of bone marrow, antecedent bone marrow transplant failure and poor graft functioning following allogeneic BMT. The gene-modified MSC, shown in the middle, will allow for the insertion into a safe harbor site of multiple genes that will then enable those MSCs, once differentiated, to be able to navigate more efficiently to sites of either primary or metastatic tumor, and then release at those sites potent cytokines, among other proteins, that are able to recruit effector T cells and other parts of the immune system known to be important in cancer tumor lysis. The potential to use iMSCs in a variety of settings that have associated with inflammation, whether that be chronic, episodic, whether it be of an autoimmune nature, is another avenue that in time will be pursued by Brooklyn. As we think about the development of gene editing and its application in vivo, the efficiency of the editing machinery will likely turn out to be very important. One, because it is overwhelmingly likely that one administration of such a product, whether it be delivered systemically to gene edit, for example, the liver, or deliver into the brain or the back of the eye, is likely to, depending upon the target, elicit an immune response. And so repetitive applications may be associated with the confound of having an adaptive immune response to the polypeptide sequences that were part of the editing machine delivered. So efficiency will be really important. The ability to knock out or correct genes of interest, of course, is of fundamental value here. We are interested in an application as it relates to familial transthyretin amyloidosis, as well as the editable mutations that are associated with one cause of blindness, mutations in the ABCA4 gene for Stargardt disease. Brooklyn has a strong balance sheet. We've raised approximately $51 million in the second quarter of 2021. We have projecting a cash runway to fund IRX2 through end of phase 2B and expand into gene editing and cellular therapies, as I've discussed. We are currently avidly cultivating BD relationships, partnerships, and other sources of capital to fuel our new pipeline. So with that, I thank you for your attention. It's good to be here at the SNN.Network Summer Virtual Event. And at this moment, I will now, for the next 10 minutes, take questions. Thank you. So one question that's been posed, what are some value catalysts moving forward? For us, some of those value catalysts will, in the IRX2 instance, pertain to the top-line data that I referenced in the first half of 2022. We anticipate that the differentiation between clinical responses, event-free survival in this case, between regimen 1 and regimen 2, one which contained IRX2, and that which lacked it, will be important because it will then delineate the ability of Brooklyn to begin to plan for a phase 3 and registrational study. With regard to the new in-licensed technologies, some of the value catalysts will be when we first enter with a unmodified IMSC into the clinic. Given what we know about the manufacturing of that product to date, given what we know about the CMC and other features, some of which have already been reviewed by the FDA, we're anticipating that we might be able to start that clinical development for the non-modified IMSC within 2 years. The gene editing for the second product and the demonstration of its effectiveness with regard to its ability to, in a preclinical model, home and ultimately kill cancer will be another value catalyst. Lastly, I think that some of the partnering relationships that we're at the early stages of exploring will also demonstrate to shareholders as well as to potential investors that we are moving forward to try to engage and ultimately have an opportunity to fuel some of the other very interesting clinical pipeline programs that I've mentioned. So I would stay tuned to those, and I think in time there will be others to fall into that catalytic context. Another question that's been posed is how would you compare Brooklyn to other companies in the same space? Well, one would be fairly obvious, and that is that we have both a cytokine in mid to late clinical development, IRX2, which is a product that may wind up being effective in a variety of combination studies. And since we know a lot about the biology of IRX2, both preclinically, non-clinically, and now clinically, there may be an opportunity for us to begin contemplating marrying that technology with one of our cellular therapies. So that's one distinguishing feature of the company now having two platforms. Secondly, we believe that our devotion to wanting to fully exploit the iPSC-derived induced mesenchymal stem cell, particularly as we contemplate serially gene editing to produce different products for different cancers and different other clinical indications, will be important as a differentiator. And then lastly, the efficiency, which I've harped on quite a lot, which is intrinsic to the way in which the synthetic RNA, when delivered with the nanolipid delivery system, the Tornado, it does not need to go through. In fact, it bypasses the endosomal pathway, which, as I'm sure all know, leads to a reduction in the expression of the transfected and encoded protein. And therefore, we think that the opportunity to take this in vivo in certain monogenic disorders for gene editing will turn out to be another part of the story that will differentiate us from other companies that are pursuing with a different generation of gene editing proteins, that which will compare, I suspect, as the clinical data arises in the minds of many who will really be believing that in vivo gene editing and the basis upon which you do it, the efficiency with which you do it, and ultimately at the end of the day, the clinical effectiveness without producing a substantial profile of AEs or SAEs will turn out to be another differentiating feature. I see there are no other questions. I'll just give the audience, if you have a few minutes, time to pose any additional ones, and if not, let me thank the organizers. It's been a pleasure to be with you, and thank you for your attention.
Ladies and gentlemen, this does conclude Brooklyn ImmunoTherapeutics presentation. Thank you for joining. You may now disconnect.